stethoscope was bad. Yeah. These are only here, so we cannot go in there. No, cannot. The auntie will get in. You understand there? That's the under ocean. That is coral reef, where do the Nemo and other fishes live. You see this one? It's my Kobe's. You see these are the
molecular biology. I studied 5 years molecular biology a long time ago already, 42 years old. And I'm a sailor. I sailed America's Cup twice as a crew member of the French team in New Zealand. And I'm in charge of the project since 15 years with the founding people, the people who founded this project at the beginning, and still in charge today. So this Tara Foundation works with a boat that is called Tara in town now. Maybe some of you went on board this week. It's an aluminium vessel, pretty small, 36 meters schooner, so 2 masts. And she's been made and built purposely to drift across the Arctic Ocean and not to go in the reef in the tropics. So life on board is kind of very hot sometimes in the tropics, but so she's made for that, and she was made for that. But since we got it, we launched her on on 11 expeditions globally. But I will speak today about the 4 main expeditions that we run over the last 10 years. It's a private funded project. It's funded by Agnès B, a fashion designer, but personally, like a philanthropist at the beginning, and still today she funds 30% of the project. And her son as well, Etienne Bourgois, who started the project with me, with us. The Prince of Monaco is supporting this project since 10 years now. We have some Swedish partners since 4 years, and Veolia since 10 years as well. And of course we work, this is private funding, and we work with public research, public scientists, basic research, which is mainly from Europe as leaders, but we also work with many others that you will see. But CNRS is one of the key players here, and EMBL, the European Molecular Biology Laboratories in Heidelberg, and CEA for genomics. All the genomics in France mainly are done by the CEA, Atomic Research Institute. So it's a non-profit funded by NESB. We have an international scientific committee. It's $3 million per year running costs about. lot of, more than that today actually, 320 days at sea every year. And the World Foundation is 22 staff. Quite a small startup, a small project. And the two missions, the one is to support scientific expedition, very focused expedition on a specific topic and try to address this question globally, always. And awareness and information about outreach, ocean literacy globally, whether it's kids, general public through the press, media, and also we are also a member at the observers at the United Nations for all the ocean-related issues, topics, governance, since 6 years now. And I already talked about that. So we decided at the beginning of this project it was really a project all about passion about the sea. Like the founders, Agnes B, she was passionate about the ocean since she was a kid, and she wanted to raise a project that is, we would be able to talk to the people about the ocean again, storytelling about that. And then, time by time, it cost a lot of money, and then what was the legacy of this project, except education, we more and more decided to run into science and try to work hard with the public scientists and basic science. And so now, at the heart of the project, we really work first on the scientific questions and what we want to do. And on top of that, we try to build storytelling, media, and outreach work. So the planet, as you can see, it is a big ball, and if you put all the air of the atmosphere in another ball, this is the size of it would be only the air we breathe on the atmosphere today. If you do the same with the ocean, with the water of the planet, it would fit into a small bowl like that. And this is the fresh water, and the smaller one is the drinkable water. So all in all the glaciers of the planet fits into such a bowl. So it's not that much. We believe it's big, very big as human beings. So big when you are looking off the coast of Hong Kong, you can't see any end. So you really feel it's really big, huge. But in fact, it's not that big. And we all know here in the room, I think that this water is a little bit under life, in marine life into it is a little bit of stress these days, especially here, but everywhere almost in the world. So as I said, we try to support science. We made sure that we provide this platform, this boat channel to the scientists across the world, and not only French, as I said. Usually, before we launch expedition, it took us 2 years to define the questions, to gather with scientists, the consortiums of people. It's really a bottom-up approach. It's not an answer to proposals. It's really a bottom-up approach all the time, which is very hard to fund. It's a very adventurous expedition in itself to do these kind of approaches, but it's very new and sometimes it's pretty disruptive the way we look at the ecosystem. Outreach, of course, education, mainly French today. It's easy to do education in France when you speak French. But once you start to fit into other systems of education across the world, everything is specific, and it takes a lot of energy, and it takes a lot of partners, local partners, to take on the content we produce in French and adapt it to the local education systems. And advocacy, we started that in 2012 when we had the chance to have Ban Ki-moon, the United Nations Secretary General, on board in New York. And he engaged us saying that you have to come on the stage, you have to come with the scientists at the UN, you have to tell about the stories he told me here. He spent two hours with us on board in New York. He was trapped on the boat. We left the harbor, and then we came back 2 hours after, so he had to speak to the scientists one by one, which is really interesting. So mainly speaking about the expeditions, I will be brief, because Chris will be talking about our ocean, the research aspect of it, and Till will speak about the Tara Pacific, which is the fourth expedition that we are doing now. That's why we are here in Hong Kong. So this was a drift across the Arctic Ocean 10 years ago. This was during the International Polar Year. And we put the boat in the ice as the only manned station in the Arctic for a year and a half. And nobody trusted we could do it. We would go to the end, and we were successful. So there was a big project called Damocles going on the thermodynamics of the Arctic. They implemented some science on board, but not enough to my taste. But it was still a successful project, I will tell you about that. Tara Ocean, coming back from the Arctic, we start to decide with Chris Boller here and with Eric Carsanti and many other people in France and in Europe to mobilize the boat for 4 years around the world to study the plankton ecosystem from 0 to a kilometer down the ocean. And Chris will elaborate on that. By doing fishing plankton, you fish a lot of plastic, so we decided to spend a full year in the Mediterranean Sea to study the plastic ocean as a laboratory, because we really know in the Med Sea the streams, we know the inputs from the rivers, and this is a lot of pressure in the Arctic over the last 20 years, demographic pressure that is, you can see in the ocean happening on plastic pollution. So in the Arctic like that, the boat is, this was when I joined the crew at the North Pole, more or less at 89 degrees north. We landed in a plane now. We were about to land at this time on the ice. We made an airstrip. So a disc boat like that drifted like a year and a half for doing 10 kilometers per day average. I think one day we did 52 kilometers with 50 knots of wind. And basically the ice we see here is drifting 10% of the wind speed, 15 degrees to the right, and it starts to drift 2 hours after the wind starts to blow. So it's really a mathematical thing. And so the boat has been built for that, thankfully. It was a success because we get the boat out and she's in Hong Kong now, so this means that all was good at this time, but still we had the big challenges because these plaques of ice are a meter average. So it's really seawater ice that is freezing. And you get to this position by going to the sea, to the ice from Russia, from northern Siberia, and you sail until you've hit the ice, you put a line on the ice, you dock to the ice, and you wait for the Arctic to freeze again. It takes 2 weeks to freeze. And then you start the polar night for 6 months. And at the end of the polar night, you arrive more or less at the North Pole, or in a degree or two from the North Pole. And all the way you can drill a hole in the ice, send CTDs, balloons in the atmosphere, try to understand dynamics and heat exchanges between the three elements, ice, ocean and atmosphere. And we run this transect all over the place for a year and a half on a daily basis. There was 10 crew on board and they've been doing that for all the journey. The drift was due to last, the drift was due to last 4 years based on mathematical models, and the boat drifted across the Arctic Ocean in a year and a half, which was much faster than we were expecting. And if you see the green one, is the only boat who did it before. The Fram was 1893. Nansen, the owner and the leader of this project, wanted to be the first man at the North Pole. This was at the time of Amundsen for the South Pole, the race for poles, and Norwegian. He missed the goal, but you should really read the book. Farthest North in a nice storybook. So you see here the prevailing winds or drift of ice. So if you stick your boat in the ice too much on this side, you go outside, you'll be ejected 6 months afterwards. If you put it here, it's a year and a half crossing the Arctic. If you go too far to the east, it's a 5 years gyre in the Beaufort Sea. So you may check it. Maybe lucky as well a little bit. So that was a successful one, anyway. And since that is the volume of ice, we often talk about the surface of the Arctic ice. Let's give you the volume evolution over the last 30 years. It was done by a US lab. The ice volume measured, estimated in at the end of September, when at the end of the summer melt, you estimate the ice volume, and this is the trend over the last 30 years. So something is happening somewhere, there, and of course you can see that before it was about 3 meters depth average, when the Russians were doing a lot of Arctic expedition with 3 meters, and now it's down to 1 meter or 80 centimeters average thickness. And more the Arctic ice is not resisting to melting. So there's no more multi-year sea ice in the Arctic, less and less actually. So more and more every year, you can see a nearly complete melt and a complete freezing before the thick ice were resisting melting on many years. So that was the story, and we're thinking now to do another drift like that in 2020. So that's in the plans for the foundation. After this project, we run the Star Ocean project on planktonic ecosystem with Eric Carsanti, Chris Bowler, Colombo de Vargas, Cecile brother was involved at the beginning of the project, and we crisscross the planet. Every dot is a sampling site of plankton at 3 depths, and Chris will explain this to you. And we came back and then launched again 6 months afterwards around the Arctic, study as well the same way. Doing this, sampling viruses, bacteria, protists, and metazoans, and also the parameters, as much parameters as we could take everywhere we sampled the ecosystem. And we did that all over every stations, over the same things. This was published, the first set of papers have been published in Science, and we had the chance to make this beautiful cover with the jewels, planktonic structures that is beautiful, in fact. And it was quite a great achievement for the team of the consortium, the scientists who did that. So at this time. And this research I can keep going in the lab so far. So we did afterwards. We did the Taramed expedition looking at plastic, how this plastic interact with the ecosystem. Try to understand which bacteria, which algae are fixed into this plastic in the range of the Mediterranean Sea. We sampled the sea from Beirut to Gibraltar, from Algiers to Marseille. 300 stations done, and every single station had some plastic in it, of course. And the concentration were more or less the same concentration you can see at the so-called continent of plastic in the Pacific Ocean. All of yes. So it's really a big concern for Europe and for Africa, and there's a lot to do there. It's very complicated because there is a lot of different cultures. The 3 main regions are around the border of the Mediterranean Sea, so it's very hard to find common understanding and common ways of moving. But it moves a little bit, and actually the Prince of Monaco is very, very well committed to this issue, and spend a lot of money into it. And we are here in Hong Kong because we work on the coral reef, and we started this in 2016. We work on it since 2014. And the project will be developed by Thiel Rotic, but it's been run again by the main partners, but also the Centre Scientifique de Monaco and Paris Sciences et Lettres, which is a university in France that is developing with École Normale Supérieure, where Chris is from. And again, it's kind of a way the same. Most of the people involved in Tar Ocean are involved in Tar Pacific. It's a way also to say that what we do now on the reef, on the coast, is pluggable, is compatible with what we've done in the offshore, on the plankton ecosystem. So connectivity could be achieved in the future, maybe. And we really study the ecosystem, again, as an ecosystem. We study the coral itself, the algae inside, the bacteria, the viruses, but also the water around. And we also try to characterize the water that's flow to the islands and flow out from the islands, which understand how it is in the context of the ocean for every reef we've been studying for 2 years. And so far it's 3,500 dives without any issues. There is some wood somewhere. I'm a sailor, so you know I'm very this is plastic. Shit. And so far it's been very successful operationally, and scientifically we are already seeing the first results are coming out of the labs, and it's pretty promising. So I'll let Thiel explain it to you. The crew was a long haul, so the white part is what we did, and where we are now in Hong Kong, and the remaining still half the planet to sail back to home. We're gonna go to mainland China, then Japan, Hawaii, US, and east and west coast, going through the French island of Clipperton. Maybe one day Mexican, but for now it's French. And then back to New York and Boston at home. So in 8 months from now, we shall be in Lorient home after 2.5 years of voyage. To give you some footage done by Pete West, one of our videographer underwater. So we sampled the seawater, the plankton above the reef. We sampled the water with specific pump that touching the reef itself, the colonies. And also so it's a lot of work, and we dive, when you're on the site, we dive 2 to 3 times a day, and usually we spend 6-7 days per sites, per islands. And so far it's about 20,000 samples collected or something like that. That's plankton net. Same as Star Ocean, but man controlled. It's like it's flying in. Many things are happening on Ocean Literacy, general public events, such as today. We have 300 kids in the morning. We have another 100 people this afternoon to visit the boat from many clubs in Hong Kong. The kids as well, and we were very well involved in the Paris Climate COP21. The boat Tara was on the Seine, at the Grand Palais nearby in the city center of Paris, with a big pavilion for ocean and climate, and we try to advocate for the importance of the ocean for climate. And 2 years before this COP21, we were asking the negotiators, what about the ocean into the climate talks, and they were saying, it's not the topic. We talk about climate, we don't talk about the ocean. Say, well, this is it's a big part of the planet, so we pushed hard with many people, with scripts, in US, with many people around the world. We gathered 85 entities together to talk on one voice for the ocean, and we also partnered with the state countries of the Pacific, island states, and in the end we managed to put the preamble of the Agreement of Paris, the word ocean. Earth and ocean ecosystem are important for the climate in the future. That was the first time, after 20 years of talks about the climate, that we consider the ocean as a key player, and key impacted, and a key player of climate change. So it was a good push, but not only us, but of course, the presence of Tara attracted a little bit of media in Paris, and we are French, but a good network in the Paris politicians and ministries. So it was pretty efficient. And some of you, I think, experienced that on board Tara in Hong Kong. And since 10 years we do that everywhere we stop by. Chris, oh, you're young, my friend. This was done everywhere. Usually when you know that, but when you do ocean cruise, you do your work at sea, and then you come back from the ocean, and you pack your things, you go to the pub, and the next day you take your plane back home, and you're happy to do it. With Star is another story. When you arrive, you have to pack your thing, but you have to stay a few days to share what you did and what you do, and explain why the ocean is so important and why you are so excited, your passion, share your passion. And this is great because the kids and the people you talk to, they are passionate about the ocean the same way you are, and then you can get engagement, engage people on this ocean that nobody really cares about. And as I said, Ban Ki-moon came in New York. He was very happy, I can see. He's happy again, less happy here, maybe. But in New York, he really stuck. He invited us to do the anecdote, and I don't have much time. Yes, he invited us at the United Nations headquarters to say thank you, bravo for what we do. 20 minutes, and take a picture with the flowers and the United Nations logo. And I asked him at the end of the meeting, "What are you doing this weekend, Mr. Secretary General?" "I don't know what I do this weekend." I don't. Look at these people like that. What I do this weekend, I don't know. Or you talk to Mahmoud Abbas and Mr. Netanyahu on Saturday morning, but then you're free. Okay? How can I come? Yes, you can come, whatever. How much people I can come? You can come with 6 people. So he showed up with his wife, his bodyguards, of course, his spokesperson, photograph, and he came with us 2 hours from the Hudson River to the East River. And he spoke to the 3 or 4 scientists who were on board at this time, one-to-one, alone. And he was very touched by this time. He told me when he left the boat, I'm very moved by what I spent on board, by what I discussed with you guys. It was not ideology, it was really facts, scientific facts, what we know, what we don't know, what is the future, what we think about the future. Really, really trying to give him the best available science, better knowledge. And so he was so touched that he came back again at the Paris COP21 conference again. So you can see Eric Carsanti here, and myself, Agnès B, one of my folks in my team, working on advocacy. And same here, Eric with shorter hairs, me with longer hair, yeah. And Mrs. Françoise Gay, which is one of the directors of the CNRS in Paris. So the future is what we're going to do in the future. We're going to do many things because the boat we'll keep doing it. We are working now on the South Atlantic program for the EU. Develop what we did on Tar Ocean, but in the South Atlantic again. Most of the consortium of the past is involved in this project, but others as well. We are thinking, Colombo Vargas is thinking about doing something in the Black Sea in the future. That's one of the planning, again for the evolution of life, so protist and plankton studies. And we also work on the new drift with a new boat that we're going to try to build in the future. We are now in the fundraising phase and the definition phase of this new boat. It's a specific boat, it's a boat that is round, like a coin. For the Arctic, to be like Tara, but round as a coin, to drift in the Arctic. It may be, seasickness may be big, may be great on board this boat. You know, like a spaceship, like it's completely round now, the one you see in the movies. We'll see, anyway. And the plan is to drift in the Arctic, to put this station in the Arctic for the next, first drift, then another drift, about 10 or 20 drifts in the future, or long-term observatory of the Arctic. That will be short on that, because you all know all this. Thank you very much, but that's a small sort of people, a small batch of people who are involved, but there's many, many more, and of course the partners. I would like to thank David Baker for having us here, and your team. Really is great from HKU to have us here, to help us a lot, to come to Hong Kong, to work together on the reef of Hong Kong at the end of the week with 3 of you scientists on board, with Till. So thank you, and thank you, Till, for you, and Cécile, for your help, for being here today. Thank you.
Okay, thank you, Romain. We will move on directly to the next presentation, and then we'll have time for question related to the interaction of the different expeditions. So the next speaker is Professor Chris Ponder from CNRS and École Normale de France. And he will really focus on Tar Ocean, as Romain introduced, and plankton diversity in the different oceans.
Something like that. Yes, thank you very much. Thank you for coming, and thank you for hosting our visit of Tara to Hong Kong, which we're very happy to be here and to share our experience and to learn from you guys as well. So I'm going to talk to you today specifically about the Tara Oceans project, as Romain has introduced to you. Tara Oceans, which is co-directed by Etienne Bourgois, the son of Agnès B, and Eric Carcenti, the scientist. Before going into the specifics about this project, just let me say a couple of words about the context of this work, which really concerned the unknown aspects of the ocean. If we think about the ocean, what it means to us, I mean, I think we all appreciate that the ocean is big and it's deep, although it's not as big as we would think. If you look at Romain's picture of the drop of water on our planet, in spite of all the work that has been done on the ocean over centuries, it's still largely unknown. But we do appreciate that the ocean protects us in various different ways. It feeds us, of course, with fish and other nutrition from the sea. The ocean absorbs excess heat, absorbs carbon dioxide, so it already performs very important jobs for us that without the ocean, the planet would be much hotter already as a consequence of anthropogenic CO2 emissions. The ocean also generates oxygen through the phytoplankton that are present in the ocean, that I'll talk about in a minute. And the ocean is also a resource not only for feed and food, but also for minerals and all kinds of other resources that we now extract from the sea. With respect to the effect of the ocean on absorbing excess heat, this just sort of a schematic of the ocean before industrial development and the current situation, with sort of looks like Hong Kong actually, sitting on the island here, and the effect of anthropogenic CO2 emissions, how that is increasing greenhouse gases in the atmosphere and causing the temperature of the planet to warm up. Thankfully, more than 90% of the heat that has been generated by human activities since the beginning of the Industrial Revolution has been absorbed has been absorbed by the ocean. Okay, so without the ocean, the temperature of the planet would already be much warmer than it is. So we need to thank the ocean for that, really, as being the planet's thermostat. And on the other hand, the ocean also absorbs carbon dioxide. It absorbs carbon dioxide through simple physico-chemical process and also through biological activity. And at the end of the day, something like 25% of the carbon dioxide that we have generated during the Industrial Revolution has already been absorbed by the ocean. So that's great news in terms of keeping the temperature down on our planet. But it's bad news because carbon dioxide and water, when you mix them together, makes bicarbonate, which is acidic. So the oceans are slowly acidifying. So put all of this together, we basically have today an ocean under extreme pressure, on the one side from climate change induced phenomena, and on the other through environmental change being mediated through different ways, pollution, not only plastic but all kinds of other chemicals. This is written in French, but you can sort of see here that we have increases in carbon dioxide in the atmosphere, increase of temperature through greenhouse effect in the atmosphere, that increases the temperature of the ocean, that acidifies the ocean, and is also deoxygenating the ocean. So we have three major insults on the ocean today: increasing temperatures, increasing acidification, and loss of oxygen from the ocean, that are hurting the way that the oceans work, that are hurting the organisms that live in the ocean, and also are affecting our coastal ecosystems such as mangroves and such as coral reefs and so on. So this is these three insults are sort of the major issues that we face today, that the ocean faces today. And this is sort of where Tara Oceans wanted to come and play a role, in particular by exploring marine planktonic ecosystems and understanding their sensitivity to climate change-induced modifications to the ocean. This was the scientific part of the project. And as Roman has already introduced you to, Tara is not only about doing hard, serious science, it's also about popularizing science for the general public, and also about educating not only young people, but also politicians, people who influence the policies that make our world spin around. So this is the context that we wanted to work in within Tara Oceans, and so we wanted to focus specifically on the planktonic organisms, which we can sort of consider as the invisible multitude of life in the ocean. If you think about ocean life, you may not necessarily think about plankton, because they're invisible, right? But actually, if you take a microscope and you look at all the plankton in the ocean and you add them all up, then you can, and you do a quick calculation, you can see that these organisms collectively represent at least 50% of the biomass in the ocean, probably even more. So these are the major players of ocean life. Okay, these microscopic organisms that we don't see, but which have all kinds of incredible functions, being at the basis of the oceanic food webs. So without plankton, there'll be no fish, there'll be no larger organisms. The photosynthetic plankton also takes carbon dioxide out of the ocean, out of the atmosphere, and brings it into the ocean, just like photosynthetic plants on land, fixes carbon into organic materials, which are then used to feed the food chains. Through photosynthesis, exactly like plants on land, these organisms generate oxygen that we breathe, which is also obviously very important for the well-being of our planet. And when these organisms die, a small fraction of them will sink to the bottom of the ocean and take the carbon with them and sequester that carbon away from the carbon cycle for hundreds, thousands, millions of years. So that makes this perhaps the most important mechanism, biological mechanism, whereby carbon can be taken out of the carbon cycle for geological periods of time. And we know through this phenomenon and other phenomenon, work performed by many people over many decades, that the plankton, on the one hand, can affect climate. They've done that in the past, during the history of life on Earth. And on the other hand, they're also affected by climate change. So there's a feedback. There are very strong interactions between the climate system and the plankton in the ocean that we need to understand in more detail. So this is the sort of context where we wanted to work in Tara Oceans. If we look now at a second, well, what is in the plankton? You can see this nice mandala of organisms here behind here, but let's just go and look at those organisms in a little more detail. One of the important components of life within the plankton are known as protists or micro-eukaryotes. These are single-celled organisms. Some of them are like algae, like chlamydomonas. Some of them are like yeasts or other kinds of fungi. You have all kinds of different morphologies that you see here. Some of them are like little boxes. Some of them are elongated. Some of them look like spaceships and so on. And these are present in very large numbers, can be present in an average litre of ocean water at up to like 10 million organisms per litre of water, okay? So that's already a large number of organisms that are present within ocean water. The larger organisms that you find, we call them zooplankton. These are organisms that can be like these copepods, little shrimp-like organisms. Other organisms with different kinds of morphologies that will typically graze on the protists, on the small organisms. So these are like the cows and sheep of the ocean, if you like, performing these important functions to transfer carbon up the food chain. These can be present at up to 100 or so organisms in an average litre of seawater. And then if we go smaller, we have bacteria, we have prokaryotic organisms, bacteria and archaea, which can be present up to 1 billion cells per litre of seawater. And then even smaller, we have the viruses and giant viruses, which can be present at up to 10 billion particles per litre of seawater. Okay? So if you add all of that up together, you get a huge amount of biomass that constitutes the plankton in the ocean. So you see that they're present in different numbers per litre of seawater. They also have very different sizes. To give you an example of what I mean here, I have this nice video where we have a zooplankton here, a copepod, with a diatom, which is a protist, sitting on it, and a bacteria sitting on the diatom here. So you can see we're going through several orders of magnitude here to go up to the bacteria that is sitting on the diatom. And we don't have the viruses here. If we had viruses, it would be a little particle on top of the bacteria. So we're going through about 5 orders of magnitude here, which, if you wanted to sample all of these organisms in a forest, it would be like sampling everything from an ant to a brontosaurus. Okay, so that's a pretty challenging technical objective, right? To sample all of those organisms. But we wanted to target all of those organisms in Tara oceans because we wanted to understand the whole ecosystem, and we wanted to do it on a worldwide scale because the ocean is one. Everything is connected, right? It may take 1,000 years for an organism sitting down here to make it up to here, but in principle, everything in the ocean is connected, and so we felt that it was important to sample the ocean in really a global setting. So as Roman introduced to you, we did 4 years of expeditions, first Tara oceans, going around the different oceans of the planet, and then subsequently going around the Arctic Circle in 2013. And at the end of all of that, we had sampled at 210 sampling sites. We had used different kinds of nets and different kinds of sampling systems to be able to catch all of those organisms, from the smallest viruses up to the largest zooplankton of a few millimeters. And at the end of the day, we had brought back something like 40,000 samples to be used for biological analyses. We also brought back a lot of other samples and a lot of other data sets that were very important to bring in the physico-chemical context of the oceans that we were sampling in. So, following that, we spent a lot of time using standardized sampling protocols, standardized analytical protocols to analyze these 40,000 samples. And the first results of our analyses, as Roman has told you already, were published in a special issue of Science already now almost 3 years ago, on the 22nd of May 2015. So we were very happy, as kind of a scientist's dream. We got 5 papers about the project, 5 papers published back to back. In this special issue, we got the front page, we got the editorial and so on, which we're obviously very happy about. We also had a huge press coverage around the world. This is the New York Times and their image of plankton on the front page. And I think in the first week following publication of that Science special issue, there were more than 500 articles published in the international press. So we're very happy with the outcome from these first results. Now, in these papers, just to give you a bit of information about what we reported in those papers, in one of them, we reported a collection of 40 million genes coming mainly from bacteria, okay, which we call the OMRGC, the Ocean Microbial Reference Gene Catalogue. This represented the largest DNA sequencing effort ever performed for the oceans at that time, in 2015. And of these 40 million genes, the majority of them are new and have unknown functions, so telling us that of the remarkable discovery that was being made in this project, and the fact that there's a lot of work to do to try and characterize what these genes are actually doing. Concerning the viruses, we reported something like 5,500 different types of viruses. Prior to this work, only 39 had previously been known. Okay, so again, showing you the remarkable discovery that was coming out of this project. And with respect to the eukaryotic organisms, the protists, we reported on the order of 130,000 genetic types of protists. This is more than 10 times higher than the number of formally described species of marine eukaryotic plankton, and about a third of them cannot be assigned to any known taxonomic group, so we don't even know where to place them in our current view of the tree of life. So those were some of the things coming out of those science papers. Just anecdotally, we now have published another resource in Nature Communications about a month ago, where we report a global ocean atlas of genes coming from the eukaryotes, so coming from protists, coming from zooplankton, and this catalogue contains 116 million genes, okay? Representing the largest catalogue of genes from any single biome ever generated by anybody to date. So these are really great resources that are ready for anybody to use who's interested to do so. So when you have all of this sort of information, you can start thinking about how you can put it all together. And one of the things that you can do when you have information about where viruses are found in the ocean, what different kinds of viruses are found in the ocean, what different kinds of bacteria are found in the ocean, what different kinds of phytoplankton and zooplankton are found in the ocean, you can throw all of that data together and you can do a very, very large compute. And you can define what we call the global ocean interactome in order to define which organisms co-occur with which other organisms, because we have such high statistical power to look at co-occurrence networks between these different organisms in all the different sampling sites. So we can define this sort of social network of the ocean, of the plankton, if you like, or the ocean's Facebook, to see what organisms interact with each other or co-occur with each other. So this is what the social network of the plankton looks like, not especially pretty, but what we have here, each dot here is a different kind of organism. In blue we have eukaryotes, in pink we have prokaryotes, and so on. And then if you see green lines connecting them, it means that these organisms tend to co-occur together. So if you find one, you will also find the other at a particular sampling site. The red lines, like here, would tend to imply that these organisms are never found together. So when you have one, you do not find the other one. Okay? So we did these kinds of interaction studies looking at organism by organism and organism by environment to see the importance of the biology of the system and the environmental parameters. And the first thing that came out of this study is that we saw that the biotic interactions were more important than the abiotic interactions, which basically means for plankton it's more important who you live with than where you live. Okay? So it's the biology that is determining the structure of the planktonic community more than the environmental parameters. So that was, we were quite surprised to see that, to get that result. Another important result that we got here that we were also surprised to get was if we tease apart those positive associations between organisms and negative associations between organisms, we saw that the number of positive associations greatly outweighed the number of negative associations, implying that cooperation is more important than competition in these ecosystems. So it's more about living together, getting along as a community, playing your part, that seems to be more important than trying to outcompete, trying to screw your neighbour and overcome them, which is a bit of a surprise, thinking about the neo-Darwinian view of evolution, which is more about, you know, survival of the fittest, competition, and so on. But this result implies no, it's more about getting along together, ensuring that that you're living within a resilient ecosystem that can deal with environmental change. So these are all interesting, you know, sort of macroecological patterns that we could find in this interaction network. But more than anything else, this interaction network is a playground to go and discover new interactions. So if you have a favorite organism, which may be this guy here, then you can go and look in this network to see who it interacts with, okay? And I'll just give you one example of that, since it's pertinent today for the coral reef talk that is coming, to give you an example of the super symbiont Symbiodinium, all right? This is the dinoflagellate, the zooxanthellae, which is the common guy in the coral reefs, which forms all kinds of symbiotic associations with all kinds of other organisms that you will recognize in these few images here. But pretty much all of these images that you see here, these interactions are always with benthic organisms. And the question that we wanted to ask, looking at this interaction network, is whether Symbiodinium is also in symbiosis in the planktonic world when it's floating around in the ocean. Is it floating around by itself, or is it in symbiosis with other organisms? And what we found from looking at this study, which was I thought it involved your brother, but Fabrice is not on the author list here. What we found here is Symbiodinium. In fact, it doesn't hang around by itself in the plankton, but it lives inside a planktonic ciliate that you see here. This hairy, single-celled ciliate organism here, which is called Tiarina, which hosts the Symbiodinium dinoflagellates inside it, in these red-blue balls inside that you can see here and also here. We can see that Symbiodinium actually lives in symbiosis with this organism when it's floating around in the plankton. We can look at the biogeography of the different types of Symbiodinium that we find and different kinds of ciliate that we find. And we can get an idea of how this ciliate is able to transport the dinoflagellate around, helping the dinoflagellate to then go and colonize new coral reefs and so on. So that's one nice example of what you can do when you use this interaction network as a hypothesis generator. And then finally, I also mentioned that we also spent a lot of time generating physicochemical information to try to help us understand more about the context of where these organisms, where these ecosystems are found in the ocean. So we measured a lot of different parameters ourselves, and we also pulled in a lot of different parameters coming in from satellite remote sensing of the ocean. For example, looking at chlorophyll from space, looking at sea surface temperature and so on, and using other very well developed resources that are being used to monitor our oceans on a daily, on an hourly basis. We could pull in all these different kinds of parameters to try to link the biology that we were looking at with the environmental, with the physics, the chemistry of the ocean. And just one of the key findings of this sort of study, to try to link the environment with the biology of the system, we found, to cut a long story short, that temperature is in fact the main driver of the structure of the microbial community. We could actually predict temperature to a very accurate extent just by looking at the plankton in the community. So this means that if you gave me a sample of seawater and I analysed the microbes that were present in that seawater, I could tell you the temperature of the water. Okay, so we've developed a very sophisticated thermometer here, most expensive thermometer ever. But that's not what's important. I think what's important here is this tells us the sensitivity of these organisms to temperature changes. Okay, and if we come back to this initial cartoon of where we have these major insults of the ocean today, including increased temperatures in the ocean, this result tends to imply that organisms are going to be strongly impacted by changing temperatures in the ocean. So I think with that story I will end, and I will just tell you that all of the data that we generate, we make public as soon as we can. We have, for example, the 150 million gene set from the ocean is now public, available to anybody who wants to use it. And we have all kinds of data sets which are publicly available, which are really there for anybody to go and study their favorite organism, or their favorite process, or their favorite gene, or whatever in the ocean. So I will end there, and thank you to the many people involved in Tyra Oceans, some of whom are shown in this photograph here, and different partner institutions and our funders as well. So thank you to them, and thank you to you for your attention. Great. And the next speaker. No. The next speaker will talk about this.
Yeah, exactly. Thank you very much, Chris, for this really deep or understanding of what ocean brings to the ocean in general.
You will now get a complete different perspective, because I have not been involved in any planning, and I have not been involved in any data analysis yet, because the whole expedition is still ongoing. So I decided that besides giving you a little more introduction in corals, I give you the perspective of just a participant on this expedition. So I'm going to show you a bit how life on board and how work on board works. And then in the end I'll try to tie that all in, what that actually means for Hong Kong, or what Hong Kong can give the Tyra expedition back. So yes, I have started a postdoctoral fellowship here at HKU at SWIMS under Dr. David Baker last November, and about a year ago I actually came off the boat in Japan after spending 2 months from Wallis and Futuna to Japan. And let's dive into corals, I would say. If we look at coral reefs, so this is a global distribution map, you see that the focus is actually somewhat off a typical world map that you would see. This is basically because the, let's say, epicenter of corals, so where we find the most different coral species and also the highest abundance, is in the coral triangle, which is around here. And most of the corals are then in the Pacific region. Corals are the largest biogenic structures on this planet. That means the largest structures that are built by organisms, and they are even visible from space. So this is a picture of the Great Barrier Reef in Australia, and you can very clearly see the coral reef. If you zoom in a little further, you see this is a picture from a coral reef in the Red Sea. But they are absolutely stunning, at least for my aesthetic sense. They are very, they feature very many colors and forms. You have fish on top, you have a lot of different shapes here. It's all very colorful, a nice place to be in, especially if the visibility is a little better than around Hong Kong. If you zoom in a little further, the organisms that are actually responsible for building those coral reefs are corals, so they are also very diverse and very different in their shape and in their color. We have foliose corals, branching corals, massive corals, but all those are actually not single animals. They are colonies of corals, and as you see here from this branching coral, every one of these tiny, let's call them balconies, actually hosts one single polyp, so one animal. They are all connected, but at the same time they are really distinct animals that live together in a colony. Each of these tiny small polyps is participating in building a calcium carbonate skeleton, and those calcium carbonate skeleton all together build these huge reefs that you can even see from space. So it's really millions and millions of tiny polyps building structures that you can see from space. It's actually pretty impressive. If you look on coral polyps, they are quite as diverse as everything else you see in the reef. In German they have been called Blumentiere, which literally translated means flower animals, and this is actually a pretty accurate description of what you see here. So these are just different species, different polyps, and they are really very colorful and very diverse. If we zoom in another step, so on the left you see one coral polyp, you see that there are a lot of small dots in there, gold brown. On the right side, at the magnification on one of the tentacles, you see them a bit clearer, and that is what Chris already mentioned before, Symbiodinium. So those are dinoflagellates, algae that live inside the coral, and they, or let's say the symbiosis between this algae and the coral, builds the foundation of the reef. So they do photosynthesis, they provide nutrition for the coral, and most of the corals would not be able to survive without this algae partner providing nutrition. You may ask now, how is it interesting for us humans? It's very aesthetic, it's very nice, it's nice to dive in there, but there are other functions as well. So it has been estimated that about a quarter of all marine life is dependent on coral reefs. That also counts for fish. And fish, of course, is a very important source for nutrition. It has been estimated that about 500 million people on this planet depend directly on coral reefs. Another very important feature that you can see over here is shoreline protection. So not only from storms or tsunamis, but there are currents and wave actions, and places that would live so close to the shore would probably have a lot more trouble if they don't have a protecting reef in front of them. If you now would go to, let's say, Central America or Central Europe, everyone would still say, yeah, I'm still safe, I'm not eating much fish, but a lot of people still want to go and swim in the sea, so tourism is a very important source of income for very many people. And then, I don't have a picture for that, but coral reefs are also a very important source for cosmetics or medicals, and most of them we don't even know yet. So all of that are very valid reasons why we should be interested in coral reefs and why we should be interested in protecting coral reefs. And I believe this has been one of the main reasons why the Terra Foundation has decided to give coral reefs a 2-year expedition. To give you the bare numbers, you heard that already partly, so the expedition is from May 2016 to October 2018, visiting more than 30 countries and 17 marinas, navigating more than 100,000 kilometers, sampling in 40 archipelagos, and there will be more than 40,000 samples coming from this period. There are 17 scientists from 8 countries on board, and there are more than 26 laboratories and institutes that partner up with Terra to make this huge effort possible. We all know about that, and we heard it already before. Actually, Chris gave a really nice introduction for that. Coral reefs are very fragile, we are losing them. Very conservative estimates are that we already lost 20 to 30%, we will lose at least another 50% until the end of the century. The main killers for coral reefs in that context are related to climate change, rising sea temperatures and ocean acidification, but there are also a lot of local threats, and they also play a big role around Hong Kong. I'll come back to that later. Pollution, overfishing, development of coastlines, and sedimentation play a big role here. The Terra approach now is to look at the health state of the coral reefs. So of course it's pretty much the same as you would go to a doctor. He's not only measuring your temperature, he's also taking a blood sample and analyzing that. So if you want to see the health state of a full coral reef, you have to do pretty much the same for a reef. And that's why the Terra Pacific expedition is sampling corals, fish, seagrass, and of course the water that is connecting all of that. So this happens at every sampling site, at every archipelago that the expedition stops, and in between it's also possible to sample plankton to get an idea. This goes along with the Terra Ocean, to get an idea what's actually happening within those huge water masses that's connecting everything. So this is the whole map. You saw it already. I came on board here in Wallis and Futuna. I went off board here in Onomichi, close to Hiroshima in Japan. And now I'm going to tell you a bit how life on board looked like, and afterwards tell you about a few examples of problems that we found. This is a part of the crew that was on board with me, so usually we are 4 coral reef scientists, 2 plankton specialists, 1 fish specialist, then we have the captain, we have a French chef, we have an engineer, deckhand, dive safety officer, and the first mate. That is the most important part, the chef. I come back to German again. We have a saying that says, translated, living like God in France. And I can tell you after 2 months on the boat, that definitely is based on food. It's really, really more than pleasant to get 3 nice meals provided by a French chef every day. And that also is really good for the atmosphere on board. Besides that, our days when we went sampling started very early, so with sunrise we had to be ready to jump in the water, and then we transferred our gear on a small dinghy and went for our dive site. And you can see that this was only fun when the weather was really great. It's pretty crowded, it's cramped with tanks, with cameras, with people, but eventually we would go there. The advantage of having a professional underwater filmer is that we get pictures like that. So this is in Tuvalu. If you know a bit coral reefs, you see that they are not really so healthy here, but it's not the point right now. This was one of our first dives. You see there are 4 people diving together. We dived in buddy teams, of course, and usually we would split up, but since this was one of our first dives, we are now first trying to identify the corals that we want to collect, to be sure that we are all on the same page and no one is sampling the wrong corals. Yeah. These are discussions underwater, it's always pretty fun. What you can see next is that we split up in buddy teams. And this is basically how we would work underwater. We have now identified a coral that we want to collect, and we use hammer and chisels and bone cutters, which are just giant clippers, to break pieces of the coral. Of course, we try to damage the coral as little as possible. You see that we are wearing lab gloves so we don't contaminate the corals. We put them then in Ziploc bags so that we can transport them back on the boat and process the samples there. That looks all nice and fine, but I can tell you as soon as there are little waves, it's not so much fun anymore. At the same time, while the coral team is underwater, there is now a second dinghy coming in, and that is bringing the plankton team in. And the plankton team, Omar has indicated that already, is now collecting water here from within, like between the branches of the coral. So we can suck those water out, it's being pumped up to the dinghy through that hose, and then that water later, I'm coming to that in a second, will get filtered and we can see what's actually living in between those coral branches. And Omer unfortunately took that surprise already from me. This would have always been my favorite job, just cruising around on the scooter through the water column and collecting plankton. I unfortunately never got to do that, but I believe it's a lot of fun. Yes, then the plankton team coming back on board, they have now between 50 and 100 liter water that they got from within the corals, and now they have a big filter party. So basically all the water goes through small filters like a coffee filter, and what sticks back on those membranes with a small pore size can now be frozen away and be shipped off the boat and analyzed in the institutes that are responsible for that. At the same time, there's also a flow cam on the boat, which is basically a camera that is connected to a really, really fine tube, and the water sample is just being sucked through, and whenever there's a particle, this flow cam takes a picture of it. And as you see here, those are tiny, mostly unicellular organisms. So you kind of automatically get firstly a count, how many are there, and you can with the pictures later go back and identify also who is actually there. So very handy and not very much labor. A little later, then the fish team, which is usually, team is a big word, it's usually just one person is going in the water later, because they usually hunt with a spear gun, and that's a bit dangerous when there are divers in the water. And they then try to catch fish, the Moorish idol that you see here, and the convict surgeonfish. They collect the same fish from all the reefs, and then they take specific samples like the gut or the ear bone or some sections of the skin. Of course, they measure the size of it, so everything will be comparable later on. While all of them are working, the coral team is actually targeting 3 coral species. This is here, Pocillopora meandrina. Here in the foreground, that is Porides lobata. And this here, fire coral, Mildipora platyphila. And it's the same 3 coral species that the whole expedition tries to sample at each spot, so we can compare it later. Now when we are done with our coral sample collection, that usually lasts about 90 minutes, we go back on the boat, and then straight away we have to preserve the samples. It's very important to do that straight away, because certain components decay very quickly. Every sample gets divided into 7 subsamples, and every of these subsamples has a different method of preserving them. This takes another 2.5 hours, and usually while we are doing that, the Terror is already going for the next site, because we don't have time ever. And I can tell you that this, with a little bit of waves, cutting coral pieces apart, putting them in small tubes, it's not my favorite occupation in the world, even though the sun was shining most of the time. After that, we tried to have some food, and then we had to go back into the water, because we had to find the precise location where on the next day we would jump in to sample the corals, and which position exactly would we find the corals for the next day. So those usually were pretty long days. We would go in the water shortly after sunrise. We would go out of the water shortly before sunset. And those days are also physically fairly exhausting. And then you can say, well, good for you that you have so long navigation times in between the islands, but someone has to label all the tubes and bags where all the samples go in. So if you have been sitting on a boat that is moving for 6 hours in a row and label the small tubes, then you also know what you have done at the end of the day. It's also not really a fun activity. Good. So let's look what we found on that transect. First, I would like to zoom into Tuvalu, because on Tuvalu, as in most of the Pacific islands, they have a big problem, which is so firstly, this is an archipelago that is only a very few meters above the sea level. We sample on 3 sites, and now one of the problems that we found on Tuvalu is trash. So none of these islands is really exporting any trash at all. 1 year back, they estimated that this landfill, which is the only one in Tuvalu, will be completely full in 3 years. So now they have 2 years to go. Of course, if you don't export any trash, also toxic trash ends up here. Of course, this toxic trash is leaking into the water and harming the corals around. This is not the only problem. What we found while diving is, of course, that there's a lot of trash just floating around, and there has been another scientific article in Science, I think now about 2 months ago, and they looked on coral reefs and on plastic trash in coral reefs, and they found that there is an increased amount of plastic trash in the reefs, then there is also a lot more coral disease. So this plastic trash, besides any other toxic substances, will also cause the corals to be sick. I find that this is actually a very powerful picture. So this is the next generation of indigenous people on Tuvalu, the future of this atoll, and one of them is having a smartphone. So if you now think about that, this will never be transported off the island, it will end up on the trash, it will leave poisonous substances in the water, which will also contribute to the corals not being able to support the island anymore. So in other words, there is a clash of culture, there is not sustainable living, and those kids, without probably even knowing it, are destroying their own future. The next example that I picked out is the Chuk Lagoon, has been previously called Truck Lagoon in Micronesia. It's an El Dorado for scuba divers that are interested in wreck diving. And what we found there is firstly that the islands in the middle are volcanic origin, so they are not quite as shallow as in Tuvalu. They have the same problem, they don't transport trash off, but I showed you that before. But what else did we found is coral bleaching. So now you probably have all heard about coral bleaching. These are pictures that are not from Tuvalu but from Samoa. Just to exemplify what happens is that here in December 2014 we have healthy, good-looking reefs. Just 2 months later, those are pretty much white, bleached, dying, and then half a year later they are brown again, but this time not because they are healthy, they are completely dead and overgrown by algae, and it is very likely that they will recover within the next decades. So coral bleaching is when the small unicellular dinoflagellates are actually being expelled from the coral, and you see what happens now. The tissue gets transparent, and you see that the white skeleton is shining through the tissue, gives the coral a much brighter, a paling, a bleached appearance. And now, I told you before, the Sympidinium is basically feeding the coral, so if they are bleached and they cannot recover in time, then the corals will starve. And this is exactly what we found in the Chuk Lagoon. We checked the temperatures because that's all available online now in databases, and we found that 2 months before we arrived there was a temperature anomaly. It was very hot there for about 6 to 8 weeks, and then we found completely bleached corals. They were still bleached, but what we also found is, like here in the front, you see that there is no tissue anymore, so not all corals completely die. They can also show partial mortality, and we had that in quite many parts of the reef. You see that there are like whitish colonies everywhere coming up, and if you have a closer look, you see here like corals that clearly have a lot of their tissue. I have to say that this, what you see here, is probably the best reef that I have seen on this whole transect. So there are still a lot of corals alive, a fairly high diversity, and I have seen a lot of reefs that look a lot worse than that, but this is the only place where we actually still saw bleached corals, and that's why I picked it. Now I would like to zoom out a bit of what I experienced on the boat and talk a bit about Hong Kong, and I believe that especially the work that is being done in Dr. Baker's lab ties in really perfectly in the scope and in what Terra Pacific is trying to achieve. If we look at Hong Kong, we have 7.5 million people living on a fairly small area. Nevertheless, we know that the marine environment around Hong Kong is very biodiverse. We have comparably many corals, we have comparably many marine species. Now, John, who's sitting actually here in the audience, is working on paleontology, on corals that are not here anymore, but also on corals that are still here. And he created this really nice map. So what you see basically is everything here that is in gray. So he's pushing big cores in the sediment and looking what corals have been there in the past. And everything that is here in gray is where he found Acropora, a branching coral that has lived there in the past, and everything that is red is where it's still alive. A little bit simplified. What he can also do with that is look how the conditions were back then from where he digs out the coral fragments. And what he found out is that basically when the corals here were still alive, they were still under a high pressure. So we have a lot of rain here, we have the Pearl River bringing in sediments and nutrients. So the corals back then even had high loads of nutrients and sediments, and this is something that is considered very harmful for most of the corals on this planet. So So we could say that the corals around Hong Kong are actually fairly tough. But there's of course another side of this story. And if you now zoom out a bit, so Hong Kong is here, the Pearl River is here, there's a lot more settlements around there, and the Pearl River is bringing a lot of pollution down that is also affecting the life around Hong Kong. And then there is another publication that came out from Dr. Baker's lab. It's called Reefs of Tomorrow, Eutrophication Reduces Coral Biodiversity in an Urbanized Seascape. Eutrophication actually means an increased amount of nutrients. And this map now shows the species richness of corals around Hong Kong. So here in the east you see that there are 60 to 65 different coral species, and while you go via orange and yellow to green, so here in the west there are only like 1 to 10 coral species, and this pretty much matches the map from John that I showed you before, but it also matches this map. So this shows dissolved inorganic nitrogen, so nutrient levels, and you see that here in the west, also where the Pearl River is coming down, the levels in red and orange are much higher than over here, and that matches basically where more corals are, the water is a lot cleaner. But it would now be a little bit too easy to say that's all coming down the Pearl River, because if you look at Tolo Harbor, you also have a gradient here, and this is clearly coming from Hong Kong. So if we now look a bit at the Tarab Pacific, of course Hong Kong is not the only place in the world where we have pollution, coastal developments and ecosystems under pressure. So maybe we have a chance now with this interconnected network of sampling, where we take samples from Hong Kong and everywhere in the world to get some idea or a glimpse what we can actually learn from other corals also to help the corals in Hong Kong. But what we definitely know already is that we have to reduce local pressure, and local pressure is overfishing, coastal development and pollution, and we have to fight climate change effectively, and that's the only way how we can help corals around Hong Kong and corals on the whole planet. And now I have another short video clip for you. I just let that sit in. I also have a lot of people to thank that especially helped Tara and helped me help Tara during the stay in Hong Kong. I have given a similar outreach talk in Germany, and people have told me, why don't you tell us what we can do to help? So in this case, I believe while we take any of your questions, it is the International Year of the Reef 2018, and there is a list of things that you can do to help protect the environment. Thank you.
Thank you very much. Jill, I think Jill, you should stay. Chris, Romain, if you want to join us here. Do you have questions for any of them? I'm sure all the three of them will be very happy to answer your question. Do you have any questions?
No, the one. Once your question is coming up on the surface, this video you saw at the end is the video we specially made for two events at the UN. The last June 8th, there was the Ocean Day, and there was a Sustainable Development Goals 14 having a big conference in New York at the UN, and we did this especially for that, and we used it another time to show it to the this Our Ocean conference that's been launched by John Kerry 4 years ago and Obama administration, and which has been taken and organized by EU last year in Malta. So we did it, this video fully opened the first day of the first speech of this event in Malta in October last year. So it was really a focus on politicians, decision makers, and local also scientists and NGOs. Any question?
How are you received in some of these countries that you visit, going to look at some of the reefs? Is it always a warm welcome? Do you have to get permits to look at these reefs? What is the, how does that work?
So how we are received, it depends. On every expedition we do, we go many 30 countries, a lot of countries, and often there is 3 to 4 countries, 10%, and it's refusing or never responding.
Hello. My name is Chiu. I'm an advanced scuba diver in Hong Kong.
Sorry. Wait for them to answer the question. I'll come back.
Yeah. What?
I'm speaking, that's why. Yes. So yeah, sometimes 10%
Just
of
talk direct.
on the way.
Without the mic. Okay,
And,
fine.
this expedition is
He's answering a question right now. Can you just wait up? He's answering a question.
I speak!
He's answering a question from someone
Okay.
else. Okay.
So this and then 10% yes. So in here we've been not able to do science, to do sampling in Indonesia, in Philippines, and in Vietnam. So it's a month for nothing, which is a month of scientists on board, full team already. And every places we had the proper scientists, local scientists working with us, already on board, ready to go, and the permit never showed up. So that's not always easy, but it's not a refusal. Sometimes they don't understand what we do, don't spend enough time, although we spent 8 months before to start to negotiate things. But and we have a big help from the French diplomacy, embassies, people, but sometimes it doesn't make it. I'm sure it's not about dealing about pollution or talking about the local pollutions. They're not afraid about that. I think it's just a question of administration, lengthy administration, bureaucracy. It kills the project sometimes. All the scientists have the same. Many boats in the world have the same issues sometimes.
I have done some coral survey for the past year, checking the coral around Hong Kong area. And I'm not sure if you have ever visited to the northeast side of Hong Kong water, where very close to Hong Kong, just across the border, there is a place in China called Daya Bay. Now that area used to be growing a live oyster farm. Now it becomes the largest container ship terminal. You know, the oyster farm has gone, and now they build a big oyster farm and terminal. So the big container ship will dump the oily water from the ship to the water where it pollutes the best Hong Kong seawater in that area. So if you have ever been there, on March 15 when you leave, you probably have to go up there and take a look at this big oil ship terminal, ocean ship terminal, whatever. So we're worried about that, and then we try to talk to China side, because the way to pollute the seawater is the growing of business, and that's what we worry about, yeah.
David, you know the place?
Yeah, I know the place. And he raises a really excellent point about cross-border communication on not just marine environmental issues, but all environmental issues. It's a pressing concern for Hong Kong people, for China people, and for our governments to work together. It's a big challenge. As far as what Terra can do, Till, what's the most northeastern site you have on your list?
Unfortunately, because of permit issues in China, the sampling around Hong Kong waters has to be a little bit compacted. So we will go on the 15th up to Crescent Island and sample there. And we hope that we get a full sample set there. So the 3 corals that we find, unfortunately we are not allowed to take any fish samples. Not sure if that would work with the turbidity here. And afterwards we go down to Lama, because we always try to have the largest distance between the sample sites that are possible. So yes,
Just like the
we'll be
Pleasant
somewhat close.
Ocean Terminal in Hong Kong on the west side, the water is so polluted that even you can get a bad smell, when you stand close to that area. So we worry about that.
Yeah. Well, I can tell you, for our
The very
research,
clean water area,
yeah,
on the east side of Hong Kong.
for our
a lot
research
we have.
at Hong Kong U, we welcome
Yeah.
observations from the public. If you see something unusual or you're concerned about something, please let
Yeah,
us
we
know.
find the best coral there, but now for another 5 or 10 years, all the coral there may be all dead.
Yeah. I share your concern. Was there another question?
Are there any specificities that you expect to see in the Chinese waters? And you've already answered a bit about the welcome by the Chinese authorities, but what's been your relationship with them so far?
There's not often that the foreign ship and the foreign people are having permits to do science in the territorial waters of China. I think it was maybe the first time. Not the EZ, but the territorial waters, it's really tough. We managed to get the permit from Beijing, the oceanographic, the Chinese Oceanographic Administration, the SOA. We managed to get the permit from the military people, because Sanya was also a military place, as you know. But I think it was too late. I mean, the order to authorize us to do things in Sanya never came down to Sanya. I mean, we showed the permits from the States in Beijing, but there was not enough. Immigration, Marine Safety Administration, Customs, and they were not aware of anything. So we waited for 8 days. We had 2 scientists from the Coral Reef Research Center of China on board the boat, waiting for 8 days with us. But it never came up, so we had to leave because we have commitment here in Hong Kong. And this is so that's why I did not mention China before in my list, because they authorized the permit, but just it never get through to the local place we were. So, but we are authorized to do sampling, the plankton sampling in the Chinese water. Then there's no more Sanya issues, local issues. So we did sample the plankton in the China Sea, in the EZ of China, not disputed EZ of China. It's a place we have to be precise on where you do things.
Any other questions from the audience?
I had to ask, in the past, is there any government or company stop Taiwan disclosing data from all the...
Disclosing data?
Yeah.
No, never.
Yeah.
But in the past, as I said, every time we do an expedition, there is people who refuse. On the Tara Ocean project, we had a no reply from Saudi Arabia. Maybe we use also, maybe it was wrong from us, we want the world, not the good pathways to get the permits, I don't know. We have no reply from Oman, because I think because the ambassador have done nothing there for a couple of months. I was a bit pissed off. We tried to negotiate everything until the end, but it was too late.